Okay, so hello everyone. Welcome to our today's Museum seminar. Um, Again, first of all, I want to thank everyone to join us during the weekend, in particular in China during the Sunday morning, and then also very special thanks to our today's speaker, Dr. Zhou Xing from Capital Medical University, Chinese Institute for Medical Research, Beijing. And then he spent the time with us today, also during the weekend. So, very brief introduction for Dr. Zhou Xing. So, um, Xin currently is assistant investigator in Chinese institutes for medical research in Beijing. He did his undergraduate study in Jilin University and then continued with his PhD training in Shanghai Institute for Biochemistry and Cell Biology, Chinese Academy of Science. Um, After that, he moved to U.S. To do his postdoc training in University of North Carolina at Chapel Hill. So from this year, he completed his training and then moved back to China, start his independent research program. So Xin's lab is really interested in develop and optimize innovative CAR T cell therapies. Try to exploring cellular engineering strategies to enhance the CAR T effectiveness against the solid tumors. Now, his lab focused on both fundamental and translational studies, addressing the key challenges of applying CAR immunotherapy in solid tumors. There's a couple of different aspects. Was mainly interested by Xing's lab. First of all, his lab is try to investigate the potential of CAR NKT cell therapy in pancreatic cancer and other solid tumors. His research leveraged both human and mouse NKT cell platform together with single cell sequencing and genetic screening strategies to evaluate potential CAR NKT cell therapy in also topic modules of pancreatic cancer and other solid tumor. Now, the second part of his lab interested is to investigate strategies to enhance CAR T cell therapy for solid tumor, mainly aim to study the signal difference between the CD28 and 4-1BB co-stimulation with the goal of modifying and developing new strategy to promote CAR T cell persistence and enhancing current CAR T therapies. Last but not least, his lab also trying to enhance the anti-tumor activity of CAR T cell by overcoming the immunosuppressive microenvironment. His lab will focus on engineer CAR T cells to target M2 macrophage in order to enhance their anti-tumor efficacy in solid tumors. So I think today he's gonna give us some overview of his lab's research general goal, and then also give us some his insight for the CAR T in the anti-tumor and in the anti-solid tumor therapies. So thanks a lot, Xing, to our platform, and we are very much looking forward to your talk.
Okay, thank you for your kind introduction from Dr. Wu, and thank you everyone to attending. So today the topic I'm going to share is to enhance the CAR-immunotherapy anti-tumor effect, especially in the solid tumor. So as you know, like I'm a fresh PI, so first slide I plan to introduce myself. My name is Xin Zhou, and I get my bachelor from the Jilin University. Then I joined SIBCB. Now they changed the name. It's called the Center of Excellence of Molecular Cell Science, Dr. Hongyan Wang's lab, where my study focuses on how that the macrophage polarization is reprogramming and how its inflammation is regulated, especially in the inflammation-related disease. Since 2019, and I go outside and to join in Dr. Dotted's lab working on the CAR T cell therapy. And I come back to China this year and join in the CIMR. I will introduce this institution later. Okay, let's go back to today's topic. what's the CAR? Of course, I'm not going to talk about the CAR in the left image. It's not an engineer conference. So the CAR is the short name of the chimeric antigen receptor, which is an artificial receptor we express on the immune cell, especially on the T cell, to help them redirect them to killing the tumor, which including sorry, maybe let me change my laser point, which including extracellular domain and intracellular domain. I will introduce these two parts during my talk. So here I prepare videos to the to the people not in this field. How does CAR-T cell work? We can see like the purple one are the tumor cell and the orange cell are the T cell. We express some receptor on the orange cell, the pink receptor. With the help of this receptor, they can kill the tumor, recognize the tumor and kill the tumor. So here is the clinical example. Here is a lymphoma patient. Before the treatment, we can see many locations are already lymphoma. But after 6 weeks CAR-T cell therapy, we can see that most of the lymphoma are already gone. So it shows very promising clinical result. So the first CAR-T cell idea is from the 1992. After around 25 years development, the first CAR-T product was approved by the FDA in 2017. So the explosion coming. Since 2017 to now, almost 8 years, there already more than 10 products are available globally from the US, from China, we can see. And but now there are still 2 things I want to mention. First, the price. We can see that almost all the CAR products, the price are still super high, millions level. It's not everyone can accept it. Second thing I want to mention is now the targeted disease are still focused on the lymphoma, leukemia, and myeloma. So far in the solid tumor, the responses are still very low. So how can we improve them? This is the very important direction in the field. So now I want to introduce you another type of the cell, NKT cell. As we all know, like we have T cell and we have the natural killer cell, NK cell. But interesting, we also need a third population, which express the T cell marker or NK cell marker. So we call it NKT cell. Compared to the traditional alpha beta T cell recognize the MHC, the NKT cell is very interesting. They recognize the CD1D. It's not this, and they not recognize the protein antigen. They recognize the lipid antigen. This difference is very important because if we want to generate off-the-shelf CAR product, the MHC restriction is very important. But the CD1D, different people's CD1D is very limited. So the different patients' CD1D is similar, so it will significantly reduce the GVHD. So the off-the-shelf product is the idea is like we can get the health donor immune cell and reprogram them in vivo to generate the CAR-T cell and infusion to patient directly. And it's many kind of advantage. For example, they can reduce the patient waiting time, and they can reduce the CAR-T cell product price. And because the CAR products are already validated, so it can give us a repeated function. Everyone knows off-the-shelf CAR product is promising, especially for the NKT cell. But NKT cell CAR product development is still very low. The major challenge is the low number. Here is the example can give you the idea how low they are. Here is the example from the mouse spleen. We can see like traditional alpha beta T cell is around 20%, but NKT cell is only 0.5. Here is the example from the PBMC. We can see that 40% are the T cell, but the NKT cell are only 0.03. So how can you expand them at a so low number and expand them at a patient use dose? It is very challenging. I also spent almost 1 or 2 years to generate the mouse CAR-NKT expansion system. We tried a different activation combination, different cytokine combination, different tissue. We tried spleen and femurs. We can see they are still not very promising. But whatever. In briefly, we generated a mouse CAR-NKT cell platform, and we can purify very pure NKT cell. Here is the CD1D tetramer and the TCR beta. This double positive is the NKT cell marker. We can very pure NKT in vitro, and we can generate the CAR receptor on in vitro compared to the T cell and the NKT cell. Interesting, in our system, we can find that the CAR-NKT cell can expand similar level like the CAR-T cell, the red and the black. Next, we want to confirm whether they can play function. So we use a very classical in vitro co-culture system. We seed the tumor cell. Here we use the B16 melanoma model, which express human CD19 antigen. After seed the tumor, and we can co-culture the CAR-T cell targeting the human CD19 CAR-T cell. And we can see in the control group, non-transduced, it means that no matter CAR-T cell and CAR-NKT cell, almost the tumor cell, which is human CD cell are positive, cell are left. NKT cell and T cell, they cannot expansion. But in the CAR-NKT cell and CAR-T cell group, we can see almost all the tumor are gone. All the cell left are the CD3 positive NKT cell and the T cell. So in vitro, we found that CAR-NKT cell shows the similar killing tumor level like the CAR-T cell. What about in vivo? So here we tried a different mouse model, the melanoma model, MT30, the colorectal carcinoma model, and blended tumor model. Interesting, we found different from the in vivo, different from the in vitro. In vivo, compared to the CAR-T cell, the CAR-NKT cell shows the best anti-tumor effect, and they can significantly enhance the mouse survival for the tumor, different tumor. What's the mechanism? First, we use the single RNA-seq to identify the microenvironment. And we found that the green one is the CAR-T cell, and the purple one is CAR-NKT cell. We found that compared to the CAR-T cell, CAR-NKT cell can significantly infiltrate in the tumor. And second, we found that compared to the CAR-T cell, CAR-NKT cell can significantly reduce the tumor-associated macrophage population, especially the M2 macrophage. We also validated by the flow and the immunofluorescence. The CD206 is M2 marker. We can see that compared to the CAR-T cell, the green one, the CAR-NKT cell can significantly reduce this population. So here we propose the first mechanism. CAR-NKT cell not only killing the tumor, they can also targeting the tumor infiltrated associated macrophage with their special invariant TCR, because this macrophage expressed the CD1D. And the second mechanism I want to mention is because our mouse model is over antigen expression. So we can use the OVA-specific tetramer to recognize the T-cell response specific targeting the OVA antigen. Interesting, we found that compared to the CAR T-cell, CAR-NKT cell can significantly promote the OVA-specific T-cell response. What's the mechanism? As we all know that the DC cell is very important for the T cell activation, so we further identify the DC cell number in the different group treatment. We found compared to CAR T cell, CAR-NKT cell can significantly enhance the DC cell number in the tumor. Further, we also validated compared to the CAR T cell, CAR-NKT cell can promote the DC cell antigen presenting ability. This mark CD103 positive DC cell was reported that can promote the DC cell antigen presenting. So here, we propose the second mechanism. CAR-NKT cell not only can kill the tumor by their CAR receptor, they can also promote the DC cell mature and recruiting more DC cell and promote their activation to help them control the tumor together. As we all know, like T cell exhaustion is a very big challenge in the T cell response in the tumor. Anti-PD-1 is widely used in clinical now. But how does the NKT cell looks like? It's unknown. So here we tested in vivo result, the PD-1 TIM3 double positive T cell exhaustion marker. We found that around 25 days infusion, CAR-NKT cell, almost half of them are showed the exhaustion like the phenotype. So it indicates that maybe we can combination with anti-PD-1. So we indeed combination with anti-PD-1. Like what we expect, anti-PD-1 combination can further enhance CAR-NKT cell shows killing tumor ability. So here we prove that CAR-NKT cell is compatible to a combination with other treatment like the anti-PD-1. The anti-PD-1 can reverse the CAR-NKT cell exhaustion phenotype to help them rebind to kill the tumor together. Here I want to introduce the second thing. Alpha-gal 3 is a very well known NKT cell agonist. Because they can, in vivo, they can activate the NKT cell. So because of this, in many preclinical studies, the alpha-gal 3 vaccine is widely used, especially for the tumor and the autoimmune disease. So the question we want to ask, whether alpha-gal 3 can combination with CAR-NKT cell. But one challenge is alpha-gal 3 half-time is very short. It's only 48 hours in vivo level degradation. So how can we deliver and make the alpha-gal 3 stable is very important. So here we collaborated with the Polo lab from Italy. We use a nanoparticle to package the alpha-gal 3. And the second way is we use the DC, in vitro BMDM co-culture the DC cell to presenting the alpha-gal 3 and to keep the alpha-gal 3 more stable. We found that with this help, the alpha-gal 3 vaccine can further enhance the CAR-NKT cell treated tumor ability. So here we propose the second combination strategy. CAR-NKT cell can also combination with other alpha-gal 3 vaccine. So here is the summary. We found that the CAR-NKT cell, even in vitro study, they didn't show the better anti-tumor effect, but in vivo, they showed the better tumor effect because they not only killing the tumor, they also targeting tumor-associated macrophage, which is the suppressive function, and they can also recruit DC cell activation and recruit more endogenous T cell response. It is very important for the long CAR T cell response. So here we propose a take-home message. I want to mention two things. First, I want to mention that the CAR-NKT cell in the solid tumor, they showed the superior anti-tumor effect. Second, they are more promising to generate off-the-shelf CAR product, which is very promising to decrease the CAR T cell price. So let's go back to today's topic. We want to focus on the solid tumor. What's the difference on the different response in the solid tumor and blood cancer? There are many challenges. We can say that in the solid tumor, they show the more suppressive microenvironment. They also show many T cell, CAR T cell especially, go infiltration in the tumor because the microenvironment is very challenging. So most of them are died very quickly. And the second thing is the compared to the blood cancer in the solid tumor, they show the more dramatically tumor heterogeneity. And then it's very challenging for the CAR T cell infiltrating in the solid tumor. At the first story I have already shown, compared to the CAR T cell, CAR-NKT cell seems like they are more promising to overcome the suppressive tumor microenvironment. Now, I want to focus on T cell dies in the solid tumor. Let's go back to the CAR T cell therapy flow. So when the patient comes, we are going to collect the blood and then get the T cell and then generate the CAR T cell in vitro. Use the cytokine, we are going to expand them at the treatment dose. Finally, we are going to infuse them to the patient. How can we ensure the sufficient quality and high quality cell product is very important. Even the CAR T cell is very good, but if they cannot expansion, it's still very challenging for to get a very good response. Now I want to introduce a background. So far, all the commercial CAR T products are based on the CD20 and or 4-1BB co-stimulator. Let's go back to the CAR product structure again, which they have to have a co-stimulator domain. So CD20 and 4-1BB are widely used. What's the difference between these two types of co-stimulator? In vitro study, we found that the most widely used CD19 CAR product, this is the human CAR product, compared to the CD20, the 4-1BB CAR, they always show the very preferred to show the big cluster phenotype. And by detecting the expansion, we found that the 4-1BB CAR shows low expansion compared to CD20 CAR. Also interesting, we found this CAR showed very high cell dies phenotype, even without antigen activation. We can see even in some donor, they showed 30% are died cell. It's unacceptable in clinical, right? How can we overcome them? By the series mechanism investigation, we found it is because CD28 and 4-1BB, they drive the different signaling. Compared to CD28, drive the PI3K, AKT pathway. 4-1BB prefer to dependent on the CHEF family. So whether CHEF can induce the cell death from the 4-1BB, so we mutated the different binding domain on the 4-1BB, which is the CHEF binding domain. And we generated 3 different truncation or mutation to abolish this binding. So we found that if you decrease the 4-1BB binding with the CHEF, it will significantly reduce 4-1BB induced cell death. So here we give the first message is 4-1BB induced CAR T cell death. It is the CHEF family dependent. So how can we use this basic knowledge to the clinical? So next, we use the in vitro co-culture system like what we did in the first story. After co-culture here, CD20, this is the tumor cell. CD3 is the T cell. After 5 days co-culture, we can see that CD20 CAR, they can significantly reduce and killed almost all the tumor. But 4-1BB CAR, there are still 20% left. But interesting, if you mutate the trap binding domain, mutation 1 and the 30-cysteine truncation, we can see that they can significantly enhance the 4-1BB CAR induced the killing. We also validated by the in vivo, by the mouse model, we can see that compared to the 4-1BB CAR, the right one, if you 30-cysteine mutation, they can significantly enhance the 4-1BB CAR induced cell death. And further, they can enhance the 4-1BB CAR cell persistence in blood. Let's go to the third story. Besides the T cell dies and the microenvironment, suppressive microenvironment, the tumor heterogeneity is also a very big challenge, which means that in the solid tumor, it's not very universal. Different subpopulations will express different antigen. For example, the orange one will express the orange one, and the purple will express the purple orange. You cannot use one kind of CAR T cell to kill all the tumor. So now the widely accepted idea is to generate a dual targeting CAR, which means that you need to express 2 or 3 CAR receptors to express on the one CAR T cell to help them to killing the different antigen. But so far, mostly we are still using the virus to express CAR receptor on the T cell. Here is the example of the dual CAR cell plasmid. We can see that it's around 4,000 BP. If you have experience on the T cell transduction, it is understandable that it is very challenging to express so high gene on the T cell at a very high transduction efficiency. So the question we want to ask, how can we make the CAR plasmid smaller? It will be very promising and beneficial to generate a dual targeting CAR T cell. So let's go back to the CAR structure again. Besides the co-stimulation domain, CAR also need an extracellular domain to recognize to take care of the binding the antigen. So far, the widely extracellular domain is coming from the antibody, which use a linker to link the VH domain and VL domain from our antibody plasmid protein. But interesting, in nature, there are some kind of the animal, like the camel, their antibody is totally different from us. We can see that in their antibody, they only have the VH domain, they're not including VL domain. And we also, at least I never hear that any of the camel had the cancer. So this idea maybe indicate that even in the VH domain antibody, they can also take a response to recognize the tumor and the passenger. So whether we can generate the VH domain based on the CAR T cell, this is the question what we want to ask. So the answer is yes. Here we compare the different, first we try the PSMA, which is the widely used prostate cancer antigen. We generated the traditional SCFV CAR and the VH based on the CAR. We can show that the VH based on the CAR can show the similar killing tumor ability. Next, we also changed to the second antigen, methicillin, which is widely expressed on the many kinds of cancer like the pancreatic cancer, ovarian cancer, and the mesothelioma cancer. In this model, we found that VH based on CAR, even shows the better anti-tumor ability compared to the scFA based on the CAR. This idea indicates that each domain can functional. What about the dual CAR? So we generated a more complicated tumor model. We express the two types of the tumor in the mouse to mimic the tumor heterogeneity. In this model, we found that the single VH based on the methicillin CAR or the PSMA CAR, they cannot kill the tumor very efficiently, but the dual CAR can show the best anti-tumor effect. So this idea shows that VH domain not only suitable to generate the CAR, especially they are also suitable to generate the bispecific CAR T cell, which is very important to overcome tumor heterogeneity. About this part, we have already applied patent, and we have already grant a commercial translational. It's a company called the Crescendo from UK. They have already set up based on the VH domain platform to generate the CAR T cell. So here is the summary. In the solid tumor, compared to the blood cancer, they have many kinds of the challenge. For example, the tumor microenvironment, we found that the CAR T cell based on the NKT cell platform showed the more promising to overcome this. To overcome the T cell dies, we genetically modified 4-1BB induced domain can significantly reduce this, can overcome it. And also use the VH domain, we can show more promising to overcome the tumor heterogeneity. The last point I want to mention is also the CAR T cell infiltration is very challenging. As we can imagine in the solid tumor, it's very solid. So the T cell is very challenging to infiltrate it. So to targeting this question, we screened many kind of the molecular, and we found that one kind of the molecular named the SWAP LCC can significantly enhance the tumor infiltration the cytokine, and also can be helpful to recruit more CAR T cell infiltrated in the solid tumor. This part of work is finished and it's under revision in the JCI journal recently, and I hope next time I can share more the detail about this part. So let's go back to the now what we are interested in. Like Dr. Wu's introduction, I go back to China and set up my own lab. So now what we are interested in, we are still want to focus on the CAR T cell and the CAR NKT cell. We want to targeting the more challenge tumor model like solid, like the pancreatic cancer and the GBM, HCC, and the breast cancer. So we want to use the orthotopic. Before we are still use the subcutaneous model. So now we want to use the more challenge model, orthotopic model to test whether CAR NKT cell anti-tumor effect. And by the way, I want to mention that about the CAR NKT cell treated pancreatic cancer, this proposal I use it to apply the very famous funding in the US, the K99, and I got a fundable score, and we got a very good feedback from the review. The review mentioned the significance, the model, and the preclinical experiment is very good feedback. Because the family issue, I decided to go back to China. So yeah, last I want to say thank you. I want to say thank you my old lab, Dr. Dotted Lab, and all the lovely lab member. And I want to say thank you all the collaboration from the different project. Here is my current lab small team. And here is our team's logo. Our duty is want to kiss off cancer. And welcome to join us at CIMR. Here is my email. At last, I want to introduce, I have my mission here. I want to introduce our institution. Our institution named CIMR, which is the total name is Chinese Institution for the Medical Research at Beijing, which is a new institution. So we want to generate a team to do, how to say that, the first class research we want to do. And here is our building under construction, and in the future in the Daxing district. And now I'm coming from the department from immunology. And here is our interested autoimmune disease, tumor immunology, and screened molecular and immunology tolerance. And here is our director now. The Dr. Mei is from the CIMR director, and Dr. Shao Chongshun is our immunology department director. And now we are looking, many faculty positions are open. So if you are interested, us, please contact me or contact Dr. Chen directly. Okay, thank you. I will stop on this slide. Okay, thank you.